Good morning, students. In this PPT, we are going to talk about Calvin cycle. So, in the last PPT on photosynthesis, we discussed how light energy is harvested to generate energy molecules, that is ATP and NADPH. That was in the thylakoids, and in this section, we'll see how these products, that is ATP and NADPH, are used to synthesize carbohydrates and other substances utilizing the carbon dioxide. So that is what is the Calvin pathway. So this pathway was elucidated between 1946 to 1953 by Melvin Calvin, John James Bashman, and Andrew Benson. They did using a tracer technique and using a radio labeled molecule, that is C14 labeled carbon dioxide. And what they did was they harvested intermediates. How did they do this? They took chlorella culture and they exposed with varying times and then varying times of illumination, and they dropped the cells after the illumination time into alcohol that will disrupt their like stop the reaction and then harvest the labeled molecule. Further, after taking out, they are separated on two-dimensional paper chromatography, and further autoradiography was done, and the products of each pathway, at different time intervals, were analyzed. So this pathway is called as Calvin cycle or reductive pentose phosphate cycle. So if they ask you to write about reductive pentose phosphate cycle, also you have to write about Calvin cycle, is also called as C3 cycle. Okay. So we'll see like how the what is this pathway. First of all, this is a cyclic process. There is taking off the carbon dioxide, and then it goes through a series of cycles, series of intermediates, and generates back the molecule which takes up the carbon dioxide, and then again it follows the same cycle. So there are three phases in this Calvin cycle: carbon fixation, that is incorporation of carbon dioxide; reduction is the second part, where there is utilization of the energy molecule to form organic compound; and further regeneration, a regeneration molecule for generating one more fresh molecule which can take up the carbon dioxide. Okay, so the cycle is something like this, and we'll see each step. The first step is carbon fixation, where carbon dioxide, that is one carbon containing molecule, mixes with 5 carbon containing molecule, that is 1,5 ribulose bisphosphate, to form a short intermediate 6 carbon intermediate, and then this 6 carbon intermediate is split down into 2 carbon molecule. So this is a synthesis reaction. It is catalyzed by ribulose phosphate carboxylase. Okay, the accepting molecule is ribulose 1,5 bisphosphate. In the presence of ribulose bisphosphate carboxylase, the carbon dioxide is taken inside with water, one more molecule of water. With the release of proton, you have 2 molecules of 3-phosphoglycerate. So one molecule of ribulose bisphosphate 1,5 bisphosphate gives out 2 molecules of the 3-phosphoglycerate. So when this reaction starts, there are 3 molecules of ribulose bisphosphate is taken, thereby generating 6 molecules of 3-phosphoglycerate. So ribulose bisphosphate carboxylase are also called as oxygenase. Further in the slides you will see the oxygenase part of the activity. It can take up carbon dioxide, or it can take up oxygen also in the active sites, adding oxygen to the ribulose 1,5 bisphosphate. So it is the abundant enzyme present on the earth. If you take the normal kinetics of the enzyme hexophosphate, or else any other enzyme, the rate of the reaction will be very high, but the rate of the reaction of the ribulose bisphosphate carboxylase is a slow enzyme, that is 3 reactions per second. That is what 3 molecules are added to 3 different molecules of ribulose 1,5 bisphosphate, and then the reaction continues. So if you take, because it is slow enzyme, so half of the protein of the leaf is made up of this protein content is made up of this enzyme, and abundant protein on the earth. And this is a very interesting enzyme because it is made up of 8 large units and 8 small units. It is called as D8L8, and then it forms this beautiful structure. The mechanism of action of ribulose bisphosphate by adding carbon dioxide is, first, the abstraction. You can see the first one, the enzyme, the group on the enzyme abstracts the proton, that is abstraction of C3 proton of ribulose 1,5 bisphosphate. The reaction, this is the reaction rate determining step. It generates a imidiolate ion that nucleophilically attacks carbon dioxide. But here it is interesting because the stroma is filled with bicarbonate also. It does not attack bicarbonate, it only attacks the carbon dioxide, resulting in a beta keto acid. The beta keto acid is rapidly attacked at C3 position by water to yield an adduct that splits the reaction, similar to aldol cleavage, to yield the two product 3-phosphoglycerate molecules. Okay, so this is the mechanism of action. Many papers, they have asked to mention the mechanism of action of Rubisco. Then you have to mention both this carbon dioxide incorporation as well as oxygen incorporation. So the first stage was the carbon fixation stage, where 3 molecules of ribulose 5-phosphate will react with 3 molecules of carbon dioxide to yield 6 molecules of 3-phosphoglycerate, which is converted into glyceraldehyde 3-phosphate, and the conversion of 3-phosphoglycerate to 1,3 bisphosphoglycerate and then to glyceraldehyde 3-phosphate is the reduction phase. Okay, so ATP is utilized here, where you have the conversion of 3-phosphoglycerate to 1,3 bisphosphoglycerate. It is carried out by an enzyme called phosphoglycerate kinase. It's a kinase reaction. Energy what is absorbed is come from hydrolysis of ATP to ADP. So there are totally 6 molecules, so 6 ATPs are utilized in this reaction. It's a phosphorylation reaction. Okay. So the next step is conversion of 1,3 bisphosphoglycerate to glyceraldehyde 3-phosphate by the enzyme glyceraldehyde 3-phosphate dehydrogenase, which uses NADPH to generate the energy, and then there is conversion of 1,3 bisphosphoglycerate to glyceraldehyde 3-phosphate, thereby releasing one of the phosphate molecule also. This is a redox reaction. The enzyme involved in this reaction is glyceraldehyde 3-phosphate dehydrogenase. So the overall reaction is 3-phosphoglycerate converting into 1,3 bisphosphoglycerate, that is converted into glyceraldehyde 3-phosphate. In presence of triose phosphate isomerase, you can have this conversion, that is isomers formation, that glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So that is the stage 1. Phase 3 is a regeneration phase. In this phase, of the six glyceraldehyde 3 phosphate produced, one of them exit the pathway to become glucose and other types of organic compounds. The five remaining glyceraldehyde 3 phosphate continues to cycle to help regenerating the substrate, that is the starting molecules ribulose 1,5 bisphosphate. Okay, one of the molecule of glyceraldehyde 3 phosphate goes to the formation of sugar, and the remaining 5 generates back the starting molecule. So the reaction synthesis, the remaining 5, that is 3 carbon containing 5 molecules, totally 15 carbon. So the starting molecule which accepts the carbon dioxide is a 5 carbon molecule, so that has to be regenerated with the absorption of energy. So the whole step is you have to follow up the numbers like 5, 6, 7, okay, and then see what I am talking. Okay, now the dihydroxyglycerol phosphate, dihydroxyacetone phosphate use okay, glyceraldehyde 3 phosphate is converted to dihydroxyacetone phosphate using the enzyme triose phosphate isomerase. These are similar to the path what you have come across, same enzyme what you have come across in glycolysis. So following this conversion from glyceraldehyde 3 phosphate to dihydroxyacetone phosphate, now there are two paths which will be taken place, that is from reaction 6 to reaction 8 and from reaction 9 to reaction 11. Okay, now this dihydroxyacetone phosphate, so first we'll concentrate 6 to reaction 6th and reaction 9. This is a kind of aldol condensation reaction in which dihydroxyacetone phosphate is linked to an aldehyde. Okay, in reaction 6 there is reverse of glycolytic reaction, that is you have formation of the by adding one more molecule of glyceraldehyde 3 phosphate, you have formation of fructose fructose bisphosphate using aldolase enzyme. And then the same aldolase enzyme, with the help of erythrose 4 phosphate, it forms pseudoheptulose 1,7 bisphosphate. Okay, now fructose 1,6 bisphosphate is like phosphate is removed with the help of fructose bisphosphatase. Now you have formation of fructose 6 phosphate. This fructose 6 phosphates, with the help of transketolase, it is converted to xylulose 5 phosphate and then erythrose 5 phosphate. Erythrose 5 phosphate along with a molecule of dihydroxyacetone phosphate, with the help of aldolase enzyme, it is condensed to pseudoheptulose 1,7 bisphosphate, which is converted to pseudoheptulose 7 phosphate by a phosphatase reaction, removing the phosphate. Now these molecules, that is pseudoheptulose 7 phosphate, is converted to one molecule of ribulose bisphosphate, one molecule of ribose bisphosphate, and one molecule of xylulose phosphate. Now the fructose 6 phosphate is also converted to one molecule of xylulose phosphate. Now this xylulose 5 phosphate is converted to ribulose 5 phosphate by the enzyme phosphopentose isomerase, which will, you can see the difference here is the reversion, or the change of position of the OH group. And ribose 5 phosphate is directly utilized by forming, ribose 5 phosphate is converted to ribulose 5 phosphate by the enzyme ribose phosphate isomerase. Now there are three molecules of ribulose 5-phosphate, ribulose 5-phosphate, and these three molecules are converted to ribulose 1,5-bisphosphate by an enzyme phosphoribullokinase involving the use of energy. Okay, now you can see here that the molecules where the energy is used is the reaction phosphoribullokinase, that is this end 13 and 12th reaction, where you have ribulose 5-phosphate formation that is converted to ribulose 1,5-bisphosphate with the enzyme phosphoribullokinase, that there 1 ATP is utilized per molecule, and then 3-phosphoglycerate converting into 1,3-bisphosphoglycerate, one more ATP is utilized, and then 1,3-bisphosphoglycerate converting to glyceraldehyde 3-phosphate, NADPH is used, and rest all the conversion there is no energy utilized. Okay, that is this reaction 1 there is 1 ATP used, and reaction 3 1 ATP is used, and reaction 4 there is a dehydrogenase reaction where NADPH is converted to NADP and phosphate. Now how this regulation or the control of, now whatever the glyceraldehyde 3-phosphate is generated, that glyceraldehyde 3-phosphate is converted to glucose, and then they are further converted to starch, and they are stored in the cells. During the day, plants satisfy the energy needs via light and dark reaction of photosynthesis. But at night, like other organisms, they must use their nutritional reserve to generate their ATP and NADPH through glycolysis, oxidative phosphorylation, and pentose phosphate pathway. Since the stroma contains the enzymes of glycolysis and pentose phosphate pathway as well as Calvin cycle, there should be a mechanism to control this Calvin cycle. That means the plants have a light sensitive mechanism to prevent the Calvin cycle from consuming this catabolically produced ATP and NADPH in a waste full cycle during the night. Okay, to waste, not to use waste those energy molecules. So to control this flux of metabolic pathway occurs at key enzymatic steps. So okay, which are like it will take a long time to reach the equilibrium. Okay, that is because they have large negative values of delta G. If you see in this table, the 4 best candidates for control of Calvin cycle are the reactions catalyzed by phosphoribullokinase, ribulose bisphosphate carboxylase, and fructose bisphosphatase, and pseudoheptulose bisphosphatase. So the catalytic active efficiency of these 4 enzymes like are being estimated, and you can see they have high value. The first enzyme, ribulobisphosphate, phosphorybillokinase, has a higher delta G value per mole, and then you can see the second enzyme also 41, and then you have the seventh one 27, and then pseudoheptulose bisphosphate 29 minus 29 delta G kilojoules per mole. So C3 plants have some limitation, that is when the stomatas are closed, there is carbon dioxide decrease, and slowly the Calvin cycle stops. Closed stomata also increases the concentration of oxygen inside, because carbon dioxide is efficiently trapped and for the preparation of organic compound, and there is slowing of Calvin cycle, and glucose is not produced. So there are some adaptations which plants have come out, like that is photo respiration limitations in C3 plants are there. And also if there is hot conditions, these tomatoes are not open because there is loss of water when it is open in arid places. So the plants have come up with C4 pathways and CAM pathways. So this is now I also mentioned that Rubisco, that is ribulose bisphosphate carboxylase, can also take up oxygen during the reactions of photorespiration. It takes up oxygen and then it generates intermediates which are similar to the when it takes up carbon dioxide. That is forming the inidiolate ion, that is enzyme groups attack the hydrogen in the C3 and abstracts it, forms a inidiolate ion. And now instead of attacking to the carbon dioxide, because the carbon dioxide is used up and there is low concentration, oxygen is added to the place. This does not form beta keto acid, but it forms something similar molecules which again undergo rearrangements. With the help of release of water, you have formation of 3-phosphoglycerate and phosphoglycolate. This is a mechanism of RUBP oxygenase enzyme. So you have the formation, if it is carboxylate reaction, you have formation of 2,3-phosphoglycerate molecules. If it is oxygenase reaction, you have the formation of phosphoglycolate and 3-phosphoglycerate. So phosphoglycolate cannot be converted directly into sugars, but it is a wasteful loss of carbon. To retrieve the carbon from it, plants must use an energy expensive process called as photorespiration. Now photo means light, respiration means consume oxygen, where the oxygen is added to the RUBP carboxylase oxygenase enzyme, which uses energy and of ATP and reducing power to generate back the molecule of ribulose 1,5-bisphosphate. So the reaction is very simple. Glycolate, what is formed? Now this okay, the action of addition of oxygen by RUBP carboxylase oxygenase enzyme onto ribulose 1,5-bisphosphate forms 2-phosphoglycolate, and this 2-phosphoglycolate is converted into glycolate by releasing one phosphate, and then you have the glycolate in the chloroplast. Glycolate is exported from the chloroplast to peroxisome, also called as glyoxysomes, where it is oxidized to glycolate by glycolate oxidase to glyoxylate. Okay, in the peroxisome it is converted to glyoxylate and a molecule of hydrogen peroxide is produced, which is acted by a heme containing enzyme catalase to neutralize the action of hydrogen peroxide. So in at times this glyoxylate is further oxidized to glycolate oxalate by the enzyme glycolate oxidase. The remainder is converted to glycine in transamination reaction and exported to mitochondria. From in the chloroplast, the RUPP carboxylase oxygenase enzyme takes up oxygen and adds to ribulose 1,5-bisphosphate, that is converted to 2-phosphoglycolate and 3-phosphoglycerate, that together with the removal of phosphate forms with the help of the enzyme phosphoglycolate phosphatase forms glycolate. Now this glycolate is transported to the peroxisome and there in the presence of glycolate oxidase it is converted to oxalate. This glyoxalate is further converted with a transamination reaction to glycine. Now, the two molecules of glycines are converted to one molecule of serine and one carbon dioxide by this glycine is transported to mitochondria where it is converted to serine with the release of carbon dioxide. Okay. This is the origin of carbon dioxide generated by photorespiration. The serine is transported back to peroxisomes where a transamination reaction converts it to hydroxypyruvate. Okay. This substance is reduced to glycerate and phosphorylated in the cytosol to 3-phosphoglycerate, which re-enters the chloroplast where it is converted to RUBP in a Kelvin cycle. The net result of this complex phosphorespiration cycle is that some of the ATP and NADPH generated by the light reaction is it is used up. So, is there any use of photorespiration? Um, so, over the time of evolution, there might be optimization of this function of the enzyme. That it is thought that photosynthesis evolved at a time when Earth atmosphere contained large quantities of carbon dioxide and very little oxygen. So the photorespiration was of no consequence. It has therefore been suggested that Rubisco reaction has an obligate intermediate that inherently auto-oxidizable. Photorespiration protects the photosynthetic apparatus from photooxidative damage when insufficient carbon dioxide is available to otherwise dissipate its absorbed light energy. So, this hypothesis was also supported by some experiments when chloroplasts or leaf cells are brightly illuminated in the absence of carbon dioxide and oxygen, their photosynthetic capacity was rapidly and irreversibly lost. So, there is some use of this pathway, but in modern time, Rubisco's affinity for carbon dioxide has negative impact on crop yield. So there are other two types of adaptation of photosynthesis, that is to dry conditions and arid conditions, that is C4 plant where they can have spatial separation and then CAM plants where they show temporal behavior or separation, and then you have the formation of the fixation of carbon dioxide apart from Kelvin cycle. So certain species of plants such as sugarcane, corn, and most important wheats have metabolic cycle that concentrate carbon dioxide in their photosynthetic cells, like totally preventing photorespiration. The leaves of these plants, like these are called as C4 cycle plants, have a characteristic anatomy that there are fine veins which are concentrically surrounded by a single layer of cells which are called as Bundle sheath cells, which in turn are surrounded by mesophyll cells. Now, this C4 cycle was elucidated in 1960 by Marshall Hatch and Roger Slack. Um, so, at first, the cycle start with the carbon dioxide which is accepted by the mesophyll cells, lacking Rubisco in their chloroplast. Uh, but they do this condensing it as bicarbonate with phosphoenol pyruvate to yield oxaloacetate. The oxaloacetate is reduced by NADPH to malate, which is exported to bundle sheath cell. There, the malate is oxidatively decarboxylated by NADP plus to form carbon dioxide, pyruvate, and NADPH. The carbon dioxide, which has been concentrated by this process, enters the Calvin cycle in bundle sheath cells. The pyruvate is returned to the mesophyll cell where it is phosphorylated to again form phosphoenol pyruvate. The enzyme that mediates this reaction is pyruvate phosphate dikinase. So this enzyme has an unusual action of activating a phosphate group through the hydrolysis of ATP to form AMP and pyrophosphate. The pyrophosphate, which is further hydrolyzed to form 2-phosphate, is equal. Energy um, is equal to consumption of 1 molecule of ATP. So carbon dioxide is thereby concentrated in the bundle sheath cells at the expense of 2 ATPs per carbon dioxide. The dark reactions of photosynthesis in C4 plants therefore consume a total of 5 ATPs per carbon dioxide fixed versus the 3 ATPs required by the Calvin cycle alone. The addition of ATP is presumably generated through the cyclic flow of electrons in light reactions. So if you compare C4 and C3 plants, C4 plants are only 5% of the terrestrial plants, and they are largely in unshaded areas of tropical region, because there they can grow faster under hot and sunny conditions than than the other C3 plants. Okay, in cooler climates where photorespiration is less of a burden, C3 plants have the advantage because they require less energy to fix carbon dioxide. So coming to CAM plants, this is also called as crassulacean acid metabolism because it was first discovered in plants of the family of Crassulaceae. Here also you have synthesis. Here there is no structural separation like mesophyll cells and bundle sheath cells. Here they are separated within the leaf only. Like desert adapted plants known as succulent employ a variant of the C4 cycle that separates carbon dioxide acquisition and the Calvin cycle in time rather than in space. As plants, they open their stomata by day to acquire carbon dioxide. They would simultaneously transpire, that is loss by evaporation, what for them would be unacceptable amount of water. To minimize this, loss, these succulent plant only absorb carbon dioxide at night when the temperature is relatively cool. This carbon dioxide in a process known as crassulacean acid metabolism, okay, by the synthesis of malate through the reactions of C4 pathway. The large amount of phosphoenol pyruvate necessary to store a day's supply of carbon dioxide is obtained by the breakdown of starch via glycolysis. During the course of the day, this malate is broken down to carbon dioxide, which enters the Calvin cycle, and pyruvate, which is used to resynthesize starch. That is, at the night time, the stomata is open and it takes up carbon dioxide and it is added to a 3-carbon, that is phosphoenol pyruvate, to form 4-carbon compound, that is oxaloacetate. Now this oxaloacetate is converted to malate by the enzyme NADPH malic dehydrogenase. This malate is further channeled to the vacuole where it is stored. Okay, so in the daytime, when the stomata is closed, when we require the carbon dioxide and to conserve water, stomata will not be open, so the light reactions have to proceed. So this malic acid comes out of the vacuole, and then the carbon dioxide is released to give malate, to give pyruvate and carbon dioxide. The carbon dioxide which is generated goes to the Calvin cycle, where it is converted into organic acids. So these are the two different steps which happens in the day and which happens in the night. Daytime, the malic acid is released out of the vacuole, and then you have generation of carbon dioxide in the chloroplast, and then you have the Calvin cycle which is getting which is done. But in the night, you have the phosphoenol pyruvate carboxylase activity, where you have carbon dioxide taken in and converted to bicarbonate. That bicarbonate is added by phosphoenol pyruvate carboxylase, that is converting it to oxaloacetate, then further to malate, and then they are stored in vacuole. Nighttime, carbon dioxide incorporation into organic acid stored in, and other molecules which are stored in vacuole. Daytime, Calvin cycle with energy from light reactions and carbon dioxide from the night storage. So in C4 plant, carbon fixation separated structurally. In CAM plant, initial carbon fixation separated temporally. So in the same cells, temporal separation is there. So the factors which are affecting photosynthesis is light intensity, carbon dioxide concentration, and temperature. If you increase the light intensity, there is a saturation maxima to which the photosynthetic activity is increased, and then it reaches saturation, and then over there, if you further increase the light intensity, there is absolutely stop of photosynthesis because the intensity is not absorbed. So the minimum amount of light that is needed to initiate this photosynthetic reaction is the light compensation point, where the minimum light intensity at which the leaf shows a net gain of carbon is the light compensation point. So people have tried to increase the temperature or lower the temperature, but as you increase the temperature, there is slight increase in the activity. If you ambient temperature, the ambient is 25 degrees, that is the optimal, and below if you go like 10 degrees, 15 degrees, you have reduction in photosynthetic activity. But if you increase the carbon dioxide concentration at lower degrees also, there is increase in photosynthetic activity. Low concentration of carbon dioxide, that is limiting the carbon dioxide diffusion. High concentration shows sometimes saturation due to other factors limiting in photosynthesis. Temperature also has certain effect, that is, as I told, the slight increase in temperature will show an increase in photosynthetic activity. So this I mean